Okay, so we discussed the other day in class and then before class partial pressure of oxygen. And if you remember these equation ish things, more sentences with numbers, that the partial pressure of oxygen follows the law of Laplace. And that's basically the pressure, the air pressure, right here, right in bright green, times the percentage of the gas in the air. And ambient air is about 21% oxygen. I don't know what it is, 21.3, 21.9% oxygen. And that gives you around 160 millimeters of mercury as the partial pressure of oxygen at sea level. Now, there's a couple of ways that you can diminish that. You can decrease the percentage, or you can decrease the air pressure. All of that is going to decrease the partial pressure of oxygen. Now, in our lungs, oxygen is around a partial pressure of 100 millimeters of mercury. This goes down a little bit because remember, your trachea, your pharynx, your bronchioles all humidify and warm the air, putting water into the air, which diminishes the percentage of the air that is oxygen because now you have all this water in there. So highly humidified air is going to go down. Also, you lose some of the oxygen percentage as it goes down to the alveoli. So at the alveoli, partial pressure is about 100 milligrams of mercury. At the tissues, which is what the cells are using the oxygen, so the partial pressure goes down to about 40 millimeters of mercury. Now, the blood that comes into the lungs then has a partial pressure of oxygen of about 40 millimeters of mercury, and the blood that leaves the lungs has a partial pressure of oxygen of around 100 millimeters of mercury. That's because at the lungs, diffusion occurs, carbon dioxide diffuses out, oxygen diffuses into the blood, and then the oxygen doesn't just go into solution, it actually binds to the hemoglobin molecule. And I can't remember if I ever put a hemoglobin molecule on here. So we can see a molecule of hemoglobin right here. And remember I mentioned the other day that you have these heme groups, which I showed you the molecular structure of. It's 4 irons that are bound to 4 nitrogens each. So you have this structure where you have a protein backbone surrounding what we call a heme group. That's a HEME group. And that consists of nitrogens with a iron which loves to give electrons or loves to reduce. So oxygen loves it, and oxygen will just come in and bind to it. So we have oxygen molecules bound to hemoglobin, 4 of them, 1 on each subunit. And these subunits can vary. Here, this is a beta chain, an alpha chain, a beta chain, and an alpha chain, which is typical of adult hemoglobin. Over 90% of our hemoglobin is made of 2 alphas and 2 beta chains. This is hemoglobin A. Now, this is the curve of hemoglobin oxygen saturation with partial pressure of oxygen in millimeters of mercury. And remember, 100 is around the partial pressure in our lungs, and hemoglobin is about 100% saturated. You get 99 point something percent saturation at 100. Now at 40, hemoglobin is about 75% saturated. And remember, 40 is the partial pressure of oxygen of a resting cell. A highly active cell may go down to about 20. But you can see at about 20 partial pressure of oxygen, hemoglobin still has somewhat of an affinity for oxygen. Oxygen and hemoglobin have an affinity for each other. So hemoglobin's still going to be bound to oxygen. But in a resting cell, it's about 75% saturated. What that means is that when you're just sitting down at your desk watching this exhilarating lecture here, your legs, your arms are all very relaxed, right? All you're going to do is drop off one of these 4, that would be 25%, leaving you a 75% saturated hemoglobin. So that's the massive buffer of hemoglobin that you have. Now, different things can modify this affinity for hemoglobin. If we come over here, remember we talked about fetal hemoglobin earlier when we were talking about the blood. Fetal hemoglobin is made up of alpha and gamma subunits, and fetal hemoglobin has a higher affinity. So at the same partial pressure of oxygen, say 40, it's 90% saturated, whereas the mother's hemoglobin is only 75% saturated. So you can modify this by putting different subunits on. Also, things that modify the affinity of hemoglobin for oxygen. pH. As the acid or acidity of the solution or as the acidity increases, pH goes down. And we talked the other day, thank you, Anna, about what could increase the acidity or decrease the pH of your blood. And that's going to be lactic acid, which is formed by anaerobic. Ooh, goodness, I don't want to write that big. Respiration. Or carbon dioxide, which is just respiration in general. Right? So, or aerobic respiration, I should just say. So you're making this carbon dioxide, it gets turned into HCO3 or H2CO3, which make the blood much more acidic, about 0.2 of a point. And what that's going to do is that's going to cause hemoglobin to lose its affinity for oxygen. So at muscles that are doing a lot of work, oxygen's going to hang on to oxygen with much less tenacity. If the temperature goes up, hemoglobin's also going to lose its affinity for oxygen. So in a muscle that's working really hard, you have a lot of work going on, a lot of friction, a lot of that energy is going to be turned to heat. Hemoglobin is going to kick off or let go of the oxygen much more readily at tissues that are producing lots of carbon dioxide or lactic acid, or that are hot. All things that happen when you exercise. Carbon dioxide itself decreases the affinity of hemoglobin for oxygen. So if you're making carbon dioxide, regardless of whether it becomes bicarbonate, it's going to cause oxygen to jump off of the hemoglobin. And a compound that is made as a byproduct of aerobic activity, diphosphoglycerate, or as they write here, biphosphoglycerate. BPG is a byproduct of aerobic respiration. And this decreases the affinity of hemoglobin for oxygen. So all of this, the important thing here is all of these graphs, they demonstrate how exercise can facilitate the release of oxygen at highly active cells. Now, so that's hemoglobin, and that's really how you get oxygen to your tissues. We talked about that the other day, diffusion basically, and then binding the hemoglobin, and then how you get oxygen dropped off at your tissues. pH goes down, carbon dioxide goes up, BPG, or just metabolites of aerobic activity, go up. Now you have the carbon dioxide you have to get rid of. And it diffuses into the blood plasma, but like I said, when it dissolves in water, it can make your blood very acidic, which can be really dangerous. So your body has multiple ways of dealing with that. Carbon dioxide is a gas, so it can diffuse right into the blood plasma. So it diffuses from the cells into the blood plasma. Much of it binds to hemoglobin and forms what's called carbaminohemoglobin. So carbaminohemoglobin is just a hemoglobin bound to carbon dioxide. The carbon dioxide can also diffuse into, oh, here's the arrow here, diffuse into the red blood cells, undergo a chemical conversion to bicarbonate, and then it's pumped back out into the bloodstream via an antiport that allows chloride to come into the cell down its concentration gradient and bicarbonate to move out into the blood plasma. Now this bicarbonate now buffers the blood plasma so it doesn't get too acidic. And another thing hemoglobin can do is actually bind protons to keep a check on the pH of the system. Now at the lungs, all of this reverses. Chloride is pumped out, bicarbonate is allowed in, it's turned back into carbon dioxide and water, then carbon dioxide diffuses out because, remember, the PCO2 of venous blood is 46 and the PCO2 of alveolar air is 40, so that's going to facilitate diffusion from the 46 to the 40, so all of that carbon dioxide just flows right out. Now the enzyme right here is carbonic anhydrase. Now our body knows exactly how much carbon dioxide and oxygen that we have in our body based on these oxygen sensors in our carotid arteries. The carotid arteries are the arteries that bring blood to our brain. And if oxygen is low, that clogs up potassium leak channels. We've seen that before. That causes the cell to depolarize. Voltage-gated calcium channels open. This is really a recapitulation of the release of insulin, if you remember that. Calcium causes neurotransmitters to release from these cells that are in the carotid artery called glomus cells. Those bind to a afferent neuron. The afferent neuron sends signals to the medulla, where you have two primary groups, the dorsal respiratory group and the ventral respiratory group. The ventral respiratory group comprises the pre-Butzinger complex, but you can just remember the dorsal respiratory group and the ventral respiratory group. Dorsal respiratory group primarily sends efferents to the diaphragm via the phrenic nerve. The ventral respiratory group sends signals for inspiration, meaning breathing in, not having an aha moment, and expiration to the pharynx, the larynx, the tongue muscles. And what this does is it allows us to make space in our pharynx for air to come in. There are also chemoreceptors in the medulla that can monitor carbon dioxide, and how they monitor carbon dioxide really is to monitor pH. Now, all of these areas in the brain, this is where it gets really fun, are controlled by the pontine respiratory group. That's the pons, so the pontine, this is just the neurons in the pons, but also paraventricular nucleus, remember that? That's your sympathetic nervous system, comes in and innervates this. The amygdala, aggression and fear, makes you gasp, makes you increase your respiratory rate, as well as hormonal input. Now, one final thing that I did want to talk about is the link, and this goes back kind of to the anatomy we were talking about Tuesday, but the alveoli Remember, these are full of these little epithelial cells here and surrounded by a very, very, very thin basement layer. So you've got the basement layer here that connects to the basement layer of the capillary. And remember, the capillaries only have that intima layer. They don't have any muscle, they don't have any connective tissue on the outside, except for the basal lamina that's at the base of all epithelial tissue. And these really become one here, and that's shown right here. This is the alveoli, and this is the capillary, the pulmonary capillary. And you can see, even though this is just a representation, an artist drawing, but you can see the close connection and no space really between here. Now, if this becomes inflamed, that's when you end up with problems. If you have, I'll try to, oh yeah, here we go. So if you have inflammation, what that's going to do is add fluid in here, and now that diffusion doesn't happen so readily. And that is why inflammation can result in hypoxia, which is a major problem with COVID-19 or SARS-CoV-2, the disease caused by that, is that you get inflammation in the lungs. These epithelial cells start to die because they're infected with virus, but it's majorly the inflammation that results from that that leads to a lack of diffusion. And there's also new evidence that I was just introduced to yesterday that proteins that are shed from the virus, I believe, can attack, or can, I guess attack isn't a good enough word, the subunit of hemoglobin, hemoglobin A. So kind of interesting there. So they're taking this out as well and leaving hemoglobin denatured. So pretty insidious. Anyway, I will see y'all in the morning.